It's identifying potential therapeutic targets in SMA through profiling of motor neuron pools that show selective resistance in human patients and mouse models.
Okay. Thank you for the opportunity to present some of our work today from the Henderson Lab. Today I'll be talking about our efforts to characterize differential motor pool vulnerability in human and mouse SMA, and our use of that in order to run a transcriptional profile to identify potential therapeutic targets. So a couple recurring themes from this meeting have been the relevance of the delta 7 model to the human disease, as dilated cardiomyopathy, other defects that may not be relevant to human, but I'll provide you with evidence that, in fact, at least at the level of differential vulnerability, the delta 7 mouse strongly recapitulates what we see in humans. And also we'll be talking about alternative therapeutic targets. As has been noted, identification of, essentially the etiology of SMA, we have a known therapeutic target in SMA has not rapidly led to the development of therapies in the clinic, so we're taking a complementary approach that we believe is needed for different therapies. So our level of analysis is focused at the level of the motor pool. So motor pool is a discrete cluster of motor neurons that innervates a single muscle in the periphery. The motor pools innervate a diversity of different muscles, so there are hundreds of anatomically distinct muscles in the periphery, and they carry out very distinct functions. This diversity is also reflected at the level of disease, which is exemplified in spinal muscular atrophy. So despite widespread flaccid paralysis in these patients, this is a typical clinical picture of a type 1 patient from a classic Dubowitz text, and despite this flaccid paralysis, you can appreciate that severe intercostal recession in conjunction with the diaphragmatic sparing that produces a bell-shaped chest that's virtually pathognomonic for the disease. Additionally, you can appreciate that the distal limbs are relatively spared, reflecting the proximal to distal gradient of the disease. Additionally, the facial muscles, the eye movements, and the sphincters that maintain continence are also relatively spared, even at end stage. So, in order to determine the pathological basis for this sparing, we performed extensive neuromuscular autopsies. So we collaborated with Wendy Chung and David Pisapia from our Department of Pediatrics and Neuropathology, and we looked at a wide variety of muscles. These are just two representative muscles. So the diaphragm on the left, you can appreciate the normal uniform architecture, the relatively uniform myofiber diameter, but in contrast on the right, the internal intercostals have severe denervation atrophy, endomesial infiltrates, and severely disordered architecture. So this reflects this is the pathological basis for the bell-shaped chest that I just showed you clinically. So we also looked in the delta 7 mouse. We used denervation analysis to correlate this with what we see in human patients. We, in order to measure denervation, we stained with vesicular acetylcholine transport marked in green, and we measured the overlap with alpha-bundler toxin to measure M plates. These are just two differentially vulnerable muscles. So the masseter on top is essentially denervated in end-stage delta 7 mouse with very little overlap between VACT and bundler toxin. In contrast, the mylohyoid down here is largely spared. It's fully innervated at end stage. So we performed extensive analysis in the delta 7 mouse. Our results are highly concordant with those of Chin-Ping Ko and colleagues, and this is just one representative example. So in red are highly denervated muscles, and in green are muscles that are relatively spared. And we were just struck by this differential vulnerability between dorsal and ventral neck muscles. And so in order to see if this was a previously unappreciated aspect in the human disease, we went back into human patients and we looked at differential neck muscles. So you can see that on the left, so these are all from the same type 1 patient, and on the left you can see that the thyrohyoid and the sternothyroid are virtually devoid of pathology. They're indistinguishable from control muscles. In contrast, the vulnerable trapezius and the deltoid on the right have severe markers of pathology. So you see fibrofatty infiltrates in the trapezius. You can even see macrophage infiltration in the deltoid. So there seems to be a remarkable correlation between the resistance in both human and mouse SMA. And this gives us confidence that we can use the mouse model, the delta 7 model, in order to elucidate the underlying molecular mechanisms of differential vulnerability and potentially other aspects of disease pathology. So, as a therapeutic strategy, if we could confer the resistance that we see on these pools onto the vulnerable pools, the therapeutic benefit might be enormous. So, this differential vulnerability, so moving back to the delta 7 mouse, differential vulnerability at the level of the neuromuscular junction is also reflected in the cell bodies centrally. So on the left is the adducens nucleus from the oculomotor complex, and you can see in end-stage delta 7 mouse, there's a full complement of cells. In contrast, the motor trigeminal, here on the right, is approximately 60% lost by end stage. So we traced the motor trigeminal back and found approximately 25% loss of cell bodies as early as P0. So this is a very severe deficit, and actually most of this deficit we found by P4. So it's an early and severe loss of cells, and this is just quantified here on the right. So moving back again, once again to the human patient, comparing the oculomotor and the motor trigeminal nerves, these are toluene blue stained semi-thin sections comparing a control to a type 1 patient. On the left, you can appreciate that the oculomotor nucleus is, the axons are largely intact. You can see a few lost axons, but in general, these axons are spared. In contrast, the motor trigeminal on the right is approximately 50% devoid of myelinated axons. This is reflected clinically by the sparing of the oculomotor neurons and dysfunction of the muscles of mastication that are innervated by the motor trigeminal that can result in, contribute to aspiration pneumonia and significant morbidity in these patients. I should emphasize that, so no matter what level we look, so whether it's at the level of a muscle, at the level of the nerve of semi-thins, or centrally at cell bodies, there's a high concordance in the pathology for individual muscles. So we can essentially look, we believe that we can look in muscles as a surrogate for individual pool pathology within the relatively morphologically homogeneous spinal cord. So this is a summary slide showing all of our data in both from human and mouse, so human on the left and mouse on the right here. The most vulnerable pools are marked in red, and the resistant ones are in green, and there's just a remarkable overlap in vulnerability between the mouse and human SMN. And so this was the basis for our profiling screen. So in order to determine the molecular basis for this, we reasoned that a pairwise comparison between, say, one vulnerable and one resistance pool, we know from previous studies would produce hundreds of gene differences. However, many of these gene differences would be related to their very divergent physiological functions or simply their anatomy. So we reasoned that by comparing multiple vulnerable and resistant pools that are paired at different levels of the neuroaxis, we would potentially eliminate gene differences that are related to developmental ontogeny, presynaptic connectivity, and simple anatomy and their divergent physiology, as I mentioned, and would potentially isolate a small number of genes that are related to vulnerability. So we also reasoned that we should look at wild-type mice because we wanted to look at intrinsic differences between motor neurons rather than dealing with the myriad of genetic effects in response to the insult of low SMN protein. So this is our experimental outlines. We labeled individual muscles. We labeled 7 resistant and 5 vulnerable muscles by using fluorophore-conjugated collar toxin B. We used laser capture microdissection, isolated individual cell bodies at P12. We ran microarray profiling and identified approximately 9 genes with a strong correlation using a strict Bonferroni correction, and that are at least 2-fold differentially regulated. So this result retrospectively validates our approach to eliminating the vast majority of genes to a much more manageable number. So if you were to take these 9 genes, for example, and compare them within any random pairwise comparison, they would fall within approximately the first 2,000 hits. So without the strategy, we would not have identified some of these genes. And so I only have time to talk about one of these genes today. I'm going to be talking about one of the most promising hits, and that is synaptotagmin 1. Here's the relative expression levels on the left, and it's much, you can appreciate that's much higher in the resistant than in the vulnerable pools, with a p-value of approximately 10 to the -5th. So what is synaptotagmin? So synaptotagmin 1 is a presynaptic active zone protein that's responsible for coordinating synchronous neurotransmitter release in precise temporal coordination with incoming action potentials at the neuromuscular junction. So it acts also with synaptotagmin 2. So it binds calcium in response to the incoming potentials. It also binds snare proteins, phospholipids, and helps to coordinate this synchronous release. So in order to rigorously test this hypothesis that synaptotagmin 1 expression segregates with pool vulnerability, we went back into the mouse and looked at motor pools that were not part of original screen. So we reasoned that if this were differentially expressed in other pools, and not only other pools, but These pools are actually, the pools we examine are actually all within the same nucleus. So these are all pools within the motor trigeminal that have related functions and mastication. So in green, the mitohyoid and the anterior digastric we found were largely innervated at end stage. The masseter was largely denervated. And amazingly, synaptogmin 1 expression, labeled here, was very high in the resistant pools, but it was virtually absent, or at least much lower levels in the masseter. We confirmed this by using adjacent sections with chat staining and also by back-labeling studies. So there's a remarkable consistent correlation between vulnerability and synaptogmin 1. We also wanted to confirm this at the protein level. So we did immunohistochemistry, so synaptogmin 1, where we expect it to execute its functions at the neuromuscular junction. So in green, you can see synaptogmin 1, and this is highly expressed in the resistant oculomotor pools, in axons, and in neuromuscular junctions. In contrast, in a more affected tibialis anterior, it's expressed at much lower levels, confirming this at the protein level. So has synaptogmin 1 been looked at in delta 7 mouse? So the study by Dale et al. Studying transport in the delta 7 mouse actually found, compared to wild type, an approximately 80% reduction in Syt1 transport to the distal axon in P11 delta 7 mice. So they ligated the sciatic nerve at P11, compared to the proximal distal components, and you can see that in the distal component there's a huge reduction in Syt1. We schematize this here, so in the wild type mice there's normal transport of Syt1 to the terminal, but in SMA there's a normal complement of Syt1, but there's its transport is selectively impaired to the neuromuscular junction. So what are the expected electrophysiological phenotypes if the localization of Syt1 to the neuromuscular junction is impaired? And these are, in fact, exactly what we see from the literature. So this is a combination of basic science studies from Tom Sudoff, along with studies looking at electrophysiology in the delta 7 mouse from Charlotte Sumner's lab, the Tavares lab, and others. So comparing the known functions of Syt, that it coordinates synchronous neurotransmitter release in response to incoming action potentials, this is impaired by approximately 50% in the delta 7 mouse at neuromuscular junctions. Syt1 is also involved in preventing asynchronous release between action potentials, and this has increased approximately 300% in the delta 7 mouse. Additionally, it's involved in presynaptic vesicle positioning. The Sumner lab has done EM studies in the NMJs of delta 7 mouse and has found approximately 30% reduction in dot vesicles. These are just three of the functions. It's also involved in actually clamping spontaneous neurotransmitter release and in determining the initial probability of release. So all these known functions of Syt are perturbed in the delta 7 mouse in a manner that's exactly consistent with what we would expect in low-level expression in the distal terminal. So to summarize our current working hypothesis, there's significant reduction of synaptogmin 1 in selectively vulnerable motor pools. We've confirmed this with in situ hybridizations and immunohistochemistry. Additionally, we've tested this in other pools. Previous studies have shown a selective inability of Syt1 transport to the distal axon, and the electrophysiological deficits that we see in delta 7 mouse are highly consistent with this function, giving us a hypothesis that reduced transport may preferentially affect the motor neurons that have low initial Syt1 expression. I know many of this dysfunction at the NMJ has been attributed to dysregulated calcium, and this may be part of the case, but it may also be the case that reduced Syt1 transport is actually a more parsimonious explanation and is highly consistent with all of the deficits that we see. So this is our current working hypothesis. So in conclusion, there's an extremely high concordance in the pathology, specifically the motor pool specificity of vulnerability in type 1 patients and the delta 7 mouse. We compared approximately 12 differentially vulnerable gene pools in P12 wild type mouse and found 9 significant differences. Among these, we're currently focusing on synaptogmin 1, this presynaptic active zone protein involved in synchronous neurotransmitter release. Our current and future studies are trying to upregulate this, either using AAV vectors or by gain of function, crossing to gain of function mutant mice with the delta 7 cross. So for acknowledgements, I'd like to acknowledge the Henderson lab, specifically Dominic Papandrea and Arden Kaplan, who've contributed to these studies. I'd like to thank George Mentis for helping with pathology, Umra Manani for our ongoing collaborations, particularly looking at these genes in less severe models of mouse, I'd like to acknowledge Wendy Chung, David Kasapi, and Jim Goldman from our departments of genetics and neuropathology that helped coordinate our autopsies for the SMA patients in the control, and of course my mentor, Chris Henderson. Thank you.
So is Syt1 expression different in the two pools in just normal animals?
Yes, so we looked in wild type animals, so there's a big difference. So our whole screen was performed in wild type animals. I should also note that Syt1 has largely non-redundant functions. So Syt1, Syt2, and Syt9 have combinatorial expression within different cells in the nervous system, and together are responsible for synaptic synchronous synaptic queries. But yes, we looked in wild type, so this is endogenous expression.
Did you find any indication for differences in expression of SMN levels itself between?
No. We have, and it's very difficult to look at the protein level. So the mRNA level, no, but the protein level, at the level of the individual pool, we're working with very small materials, so it's difficult to isolate that protein level. But the mRNA level, no.
Thank you, very interesting. We found that even within the vulnerable pools, there may be differences. For example, in the fifth cranial nucleus, we found that the muscles that close the mouth are much stronger than the muscles that open the mouth. So what does this mean? Does it mean that even within vulnerable pools there might be differences in vulnerability, or are there other explanations? For example, that some muscles just have fewer motor neurons than innervated.
So are you talking about differences within pools or within nuclei? So within the motor charge?
Nuclei, yeah. For example, are there differences in vulnerability, for example, in motor neurons that innervate in the muscles that open the mouth, and are the muscles that close the mouth less vulnerable within this nucleus?
Right, so we've looked at all the muscles in the motor shaft general, and there's an amazing differential expression. So I showed you three of the pools, the malahaya, the anterior digastric, and the masseter, which have differential expression. We've also looked at the trapezius and some other, the temporalis and some other muscles. But we actually find that the jaw closing muscles are preferentially affected over the jaw opening muscles.
Thanks.
Justin, beautiful, over here. So beautiful, very elegant work. So as you cross diseases now, because I know you do this in ALS as well, you look at resistant and susceptible pools. How concordant are those pools across diseases, say ALS and SMA? And then is synaptotagmin showing up in those screens in ALS as well? Might that be a general phenomenon, or do you think it's specific to SMA?
So for the first question, do you mean the natural history?
Yeah.
Right. Okay. So they're very different. So I think it's less well defined in ALS. Clearly, it's thought to be relative as a distal to proximal gradient versus the proximal to distal gradient in ALS. The sparing of the diaphragm is relatively unique to SMA. So we don't think. So the oculomotor nucleus and otitis nucleus innervating the sphincter muscles is spared in both. That was actually the basis for a previous screen. And one of the top hits from that screen was synaptotagmin 1. And that was so some of the previous work from Artem Kaplan and Lab I mentioned. We actually did isolate that, simply comparing the oculomotor nucleus, otitis nucleus innervating the sphincters, and generic lumbar motor neurons with the rationale of hits for ALS. So in general, there is a concordance between these, but clearly the motor specificity is very different, which simply reflects the very different etiologies. One of our hits that we found to be effective in ALS that was effective in SOD mice had no effect in delta 7 mice, and it just may reflect these differences. So we think we need to look at a larger number of pools. Thanks.
Okay. Well, I'm glad that the delta 7 is a good model. So thank you very much. So the next speaker is Amro Bonani, exploring novel determinants